And I also feel a little bit like I'm selling ice to the Eskimos, coming here to talk about aspirin with your Aspirin study, which is, obviously going to be a landmark study enrolling 18,000 odd patients. But that's in primary prevention of a number of diseases. And I'll talk to you a little bit about taking aspirin into the secondary prevention space and how it really ties in nicely with our recent advances in oncology to do with modulating the immune response. So aspirin's been around a long time. Our friend Hippocrates first used it. Acetyl salicylic acid is the fabric name and actually comes from the bark of a willow tree, so we can tell our patients it's organic. It was used around about the turn of the 1900s as a painkiller when it was used in a high dose, around 500 milligrams. And then, of course, in the 70s, we saw the dramatic results, effects on both heart and brain protection for vascular disease. Now, out of these very large randomised studies where half the patients got aspirin and half didn't, and were followed up for all-cause mortality, it was noted that there was a significant reduction in cancer incidence in patients who'd been taking aspirin. And as a result of this, there's a huge body of epidemiological work, retrospective studies, cohort studies that really document this primary prevention and also secondary prevention effect. So in primary prevention, there's data of aspirin benefit from many cancers. There's definite data that it reduces colorectal adenomas, the polyps that are the precursors of colorectal cancer. And you've seen a lot of press, particularly in the lay media, saying, why isn't everyone on aspirin? Now, the mechanism of action of aspirin is complex. It has an antiplatelet effect and an anti-inflammatory effect, as well as its antipyretic effect and analgesic effect. And all of these may be important in the pathogenesis of cancer. It's an irreversible inhibitor of COX-1 and 2, which are key in our inflammatory pathways, and they also link into the platelet aggregation pathway. And this pathway is mitigated through PIK3 kinase signalling that I will come back to in more detail later. And I won't take you through all the details of this because I want to move on to the key mechanisms. But inflammation has emerged as a major factor promoting cancer development, and aspirin will drive an in vivo shift of the tumour inflammatory profile towards classic anti-cancer immune pathways. The antiplatelet effect may prevent tumour thrombi, which may be a means of metastatic spread, but you've got a huge number of intracellular pathways that intersect with the COX-2 pathways and may also be responsible for aspirin's benefit. Now, what's really interesting and brings aspirin right to the forefront is the interaction with the PD-1 immune checkpoint pathways, and I'll come back to that at the end of the talk. So why look at aspirin with regard to colorectal cancer in particular? Colorectal cancer is the third commonest cancer worldwide, and it's actually increasing, particularly in developing countries. The fundamental shift of cancer burden towards developing countries as they reduce their mortality from infectious diseases primarily is really a concern to people interested in global health and global cancer. And by 2020, it's predicted that 70% of new cancers will occur in low-income countries. Now, in the cancer space, we live in a world of usually extremely expensive therapies, and our My particular interest is in novel therapies that may not be quite as expensive and where our neighbours in developing countries may also be able to have access to these treatments. Just going back to the data on both primary and secondary prevention, your primary prevention shows a 24% reduction in colorectal cancer incidence and 35% reduction in mortality. And you've got the meta-analysis of aspirin use post-diagnosis up on the screen, showing really a significant benefit with a hazard ratio of 0.84, about a 26% reduction. Let me tell you a little bit about how aspirin might work for secondary prevention of colorectal cancer. And to do that, you have to understand or accept the concept of micrometastases. So at the time that the surgeon removes the cancer, there are already cancer cells that have spread to distant organs that we cannot pick up with the resolution of our current imaging. That the concept of adjuvant therapy, if we give chemotherapy at that stage, down the track less people will develop secondaries, so we believe we've eradicated these micrometastases. But we still have to treat a lot of people to benefit a few, and that's where biomarker selection would be extremely helpful. So for 100 patients with stage 3 colorectal cancer, that is cancer that has lymph node metastases that are removed at surgery along with the primary tumour, we have a number of patients who are cured with surgery alone, who currently get chemo but didn't need it. The unfortunate group in the middle who recurred despite having had surgery and chemotherapy, and so the chemotherapy didn't actually help, or at least didn't do enough. And then we have the small number of patients who are cured because of the chemotherapy, either by one drug, the patients in red with fluoropyrimidine, or combination drugs. If you look at an unselected population and look at patients who took aspirin after their diagnosis of colorectal cancer, not to prevent it coming back for other reasons, and in the US in particular, aspirin is used where we in Australia would use paracetamol. It's used as an anti-fever much more than we use it. And if you look across an unselected population, you will see a small but significant benefit in both colorectal cancer, specific survival and overall survival. I'll talk you through the PIK3 kinase story, and many of these biomarkers were identified from this very large combination of cohort studies, the Nurses' Health Study and the Health Professionals Follow-Up Study. So these were very large cohorts of supposedly reliable people. We don't lie about our health or our diet or our drug intake. So we were, nurses and health professionals were picked as reliable people, and they were followed, over 100,000 people were followed and had really documentation of all the health problems that they encountered. And if you look at the cohort of the 964 patients who ended up developing bowel cancer in those studies, these investigators went and retrieved the samples and did a whole lot of biomarker studies on the tumour sample, including all of our standard predictive biomarkers. And what they found, and this is just a cartoon of the PIK3 kinase pathway, which is really one of the most important pathways in cell growth, differentiation and cell survival, is that if you look on panel A at the colorectal cancer specific mortality, if you have a mutation in exon 20 of the PIK3 kinase gene and you didn't use aspirin, you have a probability of death up around 0.3, and if you use aspirin, you have almost no chance of dying of colorectal cancer, which is pretty stunning for a treatment that wasn't actually given for the disease. If you have wild type PIK3 kinase in panel B, you can see that there's no effect of the aspirin. And similar effect is seen with overall mortality if you have a PIK3 kinase mutation or wild type. So I'll lead you on to the next bit of evidence in this story, and that comes from our Victus study, which was a study of Vioxx. And in Australia, through our Australian Collaborative Colorectal Trials Group, the AGITG, this study was taken up with great gusto, came from our collaborators in Oxford, and this was a study of adjuvant non-steroidals following colorectal cancer, because non-steroidals were far sexier than using aspirin, and everyone thought that this would really may be an effective adjuvant therapy. So patients were enrolled and either got rofecoxib or placebo, and you will all recall, but perhaps not the timing, about 9 months into the trial, the drug was withdrawn overnight, stopped production in all parts of the world because of underreporting of cardiac and vascular toxicity. And so thanks very much, there goes our study. But we did have about 9 months of patients enrolled onto the study. And because they were being enrolled onto a non-steroidal study, data was also collected on aspirin use. And what we found was the rofecoxib seemed to not make any difference, and we couldn't find a biomarker. But if you look at aspirin use and you look at PIK3 mutation in this data set, you get exactly the same data as in the retrospective cohort. Almost no relapses in the mutant PIK3 kinase group. Now, I'll take you through our current study, but we did some modelling to show that even if aspirin had only a modest treatment benefit, aspirin is likely to be cost effective. So I have to always defend oncologists against using up our health budget on very expensive drugs. So this is our ASCOLT study. I'm one of the international principal investigators. It's patients with stage 2 or 3, so early colon cancer, completely cured by surgery except for those with micrometastases, and they're randomised to either aspirin 200 milligrams. Happy to answer questions on the dose, I'll come back to it, but it was a compromise between not being too low with our anti-inflammatory dose versus our anti-thrombotic dose. Patients are randomised to aspirin versus placebo for 3 years. And this study is recruiting about 1,700 patients, mainly through the Asian region. Australia's the largest Caucasian-based population in this study. It's also a landmark for showing that countries in the Asia-Pacific region can collaborate on important studies. And we're the first of the adjuvant aspirin studies. There's a number of other studies that took a long time to start. Some of them cover other cancers apart from colorectal cancer. There are a couple of studies that are just looking in PIK3 kinase mutations, tumour mutations, and we do have an aspirin trialist collaborative group that has agreed to pool data for subsequent meta-analyses and also to share our translational studies. So I lead the translational program for the ASCOLT study, and we are doing both discovery and validation to look at predictive markers of aspirin benefit with 3 main aims: de novo discovery using some cell lines, human colorectal cancer cell lines, looking through candidate biomarkers from the literature, validating them in the ASCOLT, and then further validation in the Victus study, as well as the Scott international study. The principal biomarkers, as I've mentioned to you, are PIK3 kinase, which occurs in about 15% of colorectal cancer, perhaps a little bit lower in the not too low, because having adequate power in these studies to test your biomarker is an issue. We're also looking at COX-2 overexpression by immunohistochemistry. There's a whole range of SNPs that we are looking at as well. Some analysis of HPGD expression. We're looking at BRAF mutation, and we're looking at lymphocyte reaction and the immunoscore. And we've just added a couple to do with PD-L1 expression, and I'll come back to that. But obtaining enough patients in your biomarker group is a real problem for us in clinical trials, no different to stroke. Cancer is becoming a series of rare diseases, so I endorse what Richard said about rare diseases. But we think we will have the power to validate PIK3 kinase as a biomarker if it is around the 15% prevalence mark. Let me bring you to the story of colorectal cancer and immunogenicity, and particularly the role of deficient mismatch repair. So as you'll know, DNA is damaged constantly, and our cells have very efficient mechanisms to repair different types of DNA damage. Mismatch repair repairs base incongruities between the different strands. And the proteins, there are 4 human DNA mismatch repair genes that can be affected, either germline, giving rise to HNPCC, hereditary non-polyposis colon cancer, or more commonly somatic mutations, which occur in about 15% of sporadic colorectal cancer. Now, microsatellite instability is just a marker. It's an easy detection marker for deficient mismatch repair, and it does not of itself cause cancer, and sometimes there's confusion there. Now, depending on whether you have a sporadic or a germline mutation of your mismatch repair genes, you get a different phenotype. And people with HNPCC get a number of cancers and have a mutation in their gene sequence, whereas sporadically it's mainly an epigenetic phenomenon of hypermethylation of the MLH1 promoter. Microsatellite unstable colorectal cancers look nasty. They look very immunogenic. They have a lot of what are called TILs, tumour infiltrating lymphocytes, and they're often poorly differentiated, but surprisingly they do better. So the body is actually recruiting the immune system. We can see it on the histology, and these patients have a lower risk of relapse. Looking at the mutational load of colorectal cancer, we don't have a particularly high mutational load, except for those cancers that have microsatellite instability, or as I said, deficient mismatch repair. And if you look at our new generation of checkpoint inhibitor drugs, the PD-1 drugs that have had such remarkable, really have been game changers in the last couple of years in oncology, and one of them was listed on the PBS today for lung cancer, and we've had thousands of calls from patients. But if you look at PD-1, it doesn't work in colorectal cancer, except for cancers with mismatch repair deficiency. And in patients with mismatch repair deficient colorectal cancer, the hazard ratio for death was 0.22 using this trial of pembrolizumab. And if you look at the whole genome sequencing of the tumours in the patients who responded, they had a mean of about 1,800 somatic mutations compared to about 73 in mismatch repair proficient tumours. And this is the dramatic data in both mismatch repair deficient colorectal and non-colorectal cancers, showing the impact of the PD-1 checkpoint inhibitors. Now, I haven't gone completely crazy. I am going to bring aspirin back into this, and this is our trial that hopefully is looking like it will get funded, some pharmaceutical support. The whole idea of those of us who are dealing in cancers that used to be in the plenary sessions of our conferences and are now relegated to the back, while tumours like melanoma, who used to be in the back room with no one attending, grab all the plenaries. So, you know, Mark Shackleton, who I know will start here tomorrow, and I'm very much looking forward to working with him. The melanomas docs really lead the pack at the moment. But we are looking at trying to make non-immunogenic cancers immunogenic. And one of the ways we're doing this and we're testing with this trial is using a combination of both radiation, which we know induces an immune response, plus a PD-1 checkpoint inhibitor, plus aspirin. So we know patients who take aspirin during radiation actually have a better survival. We know that in preclinical models, if you combine aspirin and PD-1, you get a dramatic shrinkage in tumour control with no effect where you have one or either alone. And this is the schema that we hope we'll be able to try. This is a very landmark trial we have, because you have patients who have their cancer still in situ, they're having neoadjuvant treatment, and we can do serial biopsies. And actually the endpoint of this study is translational endpoints. And the clinical endpoints are secondary. And if we can get industry to start funding trials with translational endpoints and not rushing into very large randomised phase 3 trials where really we might get a negative result because we're not picking up on the subgroup of patients where the treatment works. So I think this is the way forward. And just to bring this all together, there's a recent publication, June the 1st, looking at those very same group of the cohorts of the nurses' health and health professional studies, simply by staining their tumour for the PD-L1 ligand, PD-1 L1 ligand. You can see that the patients who used aspirin and had a low expression of the PD-L1 receptor had much better survival if they regularly used aspirin. So I think aspirin is going to link the immunogenic pathways, the PD-1 inhibitors, and trying to make colorectal cancer immunogenic. The final twist in the story is that we know that melanoma patients who have mutant BRAF, which is a particular pathway, we know that those cells are also poorly immunogenic. So these mutations are turning off and not recruiting the immunogenic pathway to enhance anti-tumour effect. So thank you very much. I hope I've convinced you that if you don't take your aspirin before you have your cancer, you certainly should after you have your cancer. And just to acknowledge Oliver Sieber's laboratory at WeHi, who's my main collaborator for the translational study, the ASCOT investigators, and some funding from the Perpetual Foundation. Thank you.